Hi Life Sciences Biology students, I hope that you are gaining lots from this range of prac skill videos. Now we're going to be looking at accuracy versus reliability when you are doing a practical. Now often students get very confused between the two parts, but I think it's pretty clear and hopefully this video will make it a lot clearer for you. Okay, so let's first look at the word accuracy. How can your results be accurate? Okay, now that means how did you work carefully in order that your controlled variables were controlled? So when I say how do you work accurately or how do you work carefully, I'm asking you about the controlled variables. What did you do to make sure that the controlled variables were controlled? How did you make sure that you put 20 ml of liquid into each test tube? How did you make sure you measured exactly 5 ml of glucose? What did you do? How did you work carefully to make sure your results are accurate? Okay, so now we're just going to look at the one experiment. Okay, I've changed it a little bit to determine which substances, apple juice, lemon juice, and egg white contain glucose. Okay, so the first thing is when you read your experiment, it will say to you, add 10 ml of each substance to each test tube. So here are your test tubes. Okay, A, B, and C, and you now need to add 10 ml of each of these substances to each test tube. How can you do that accurately? How are you working carefully? So there are two options. Either you can have a measuring cylinder or you can have a syringe. So let's work out how can you work carefully with each of those. So a measuring cylinder is much easier actually than a syringe. All you do is you pour the liquid into the measuring cylinder and you've got your gradations or you've got your values, and it's very important to avoid the error of parallax. So when you're looking, you will bend your knees and you make sure that you are looking at the value on eye level. So if you're measuring 10 ml, remember the meniscus. Okay, here is your measuring cylinder. If that is the 10 ml mark, the bottom of the meniscus must be on the 10 ml mark. That is how you do that. Okay, right. So that is if you're using a measuring cylinder. Now if you use a syringe, when you suck things up, obviously you can get air bubbles in, you might not be getting an accurate result. So when you use a syringe, you suck up the liquid using the syringe, and you must always suck up more than the desired amount. So let's say if you've got 10 ml, suck it up to about 12 ml, somewhere around there. Overfill the syringe. And that is because you will have air bubbles. So it's important to get rid of those air bubbles, otherwise you're not measuring 10 ml and that will not be a controlled variable. So how do you get rid of air bubbles in a syringe? I'm sure you've all seen doctors or programs. They invert the syringe, they flick it a little bit, and those air bubbles then come to the top. Those air bubbles have to be expelled. So now you'll push the syringe, the plunger, you'll push or depress the plunger to remove all those air bubbles. It still might not be at your 10 ml mark. So now you might have to depress it a little bit more until the tip of the syringe is touching the 10 ml mark. And that is how you can make sure that your liquid volumes are identical. And then sometimes you will need to measure the mass of a substance or the volume. So let's say we are measuring glucose, we're making the positive control, for example. There are two options. Either you can use a mass meter or a scale. How can you work accurately with a mass meter or a scale? Well, obviously before you add anything, you need to zero it. Okay, so you push the button, I think it says 10 usually or something else, and you need to zero it so that it's exactly the amount, and then you'll add your substance. If you are measuring with a spoon, often you'll use a medicine spoon or any type of spoon that will measure 5 ml, 10 ml. You can't just lift up a little bit of the glucose powder. You need to make sure that the entire spoon is full with the glucose powder. So again, you overfill the spoon. Once you've done that, you take a blunt object. For example, you can take the back of a knife, or if you've got a ruler, you can use a ruler, and then you will smooth it off so that it's exactly 5 ml or 15 ml or whatever your value is. Okay, sometimes you'll have to look at temperature. This is not in this particular example, but if you have to work out how to work accurately to work out the temperature. Now, this is often people do this incorrectly. They just put the thermometer into the solution or into the water, they look at it and say, oh, it's 43 degrees. But you need to put the thermometer in and give it time for the mercury to rise so that it can actually get to the correct temperature. So, about 30 seconds, you mix it in and you'll get the accurate temperature. Again, because you're looking at a value, you need to make sure you're on eye level with that value to avoid the error of parallax. Okay, so that is the third one, that is temperature. And also, you must be careful, if you have lots of different solutions or lots of different powders, you must prevent cross-contamination. So that again is working carefully to ensure your results are accurate. Because if you're mixing glucose powder with starch powder and anything, or anything else, then of course you've got cross-contamination and your results will be completely inaccurate. So those are the ways that you can work carefully to avoid mistakes with your controlled variables. Okay, let's look at reliability. So you've done this experiment and you've got that apple juice contains glucose and lemon juice and egg white don't contain glucose. Okay, fine. But how can I ensure that those results are reliable? There is one standard way, and that is to repeat the experiment. If you repeat the experiment and you get the same results, then you know your results are reliable. It's good to do that, especially with quantitative results, because you have values. And if you do if you do 5 of each experiment, then you can get an average of the values, and then your results are much more reliable. Also, if you wanted to do this, you could have had 3 test tubes with apple juice, 3 test tubes with lemon juice, and 3 with egg white, and you could have done them all together, and then that would give you more reliable results. Do not ever include another variable. Never say, okay, let's do orange juice and other juices, because that is changing the experiment completely. To make an experiment reliable, you do more of that same experiment. And another thing that can also make an experiment more reliable, if there is no control, you can add a control. So to make this experiment more reliable, you'd add the positive control of glucose and water, pure glucose, which you know will have a positive result, and you can add water, which you know you have a negative result, and then you can compare, and that makes it more reliable. Right, so that is the difference between accuracy, reliability when you're doing experiments.